Hello and welcome to this module on electrophoresis theory. Before I begin discussing electrophoresis theory, I want to briefly introduce the power supply and the electrophoresis chamber because an understanding of both the power supply and the electrophoresis chamber to some degree is important to understanding the electrophoresis theory. First, I want to discuss the power supply. So by definition, the power supply will convert electrical current to a specific voltage and current. So here to the top left, you can see a picture of a power supply. You can see that the voltage can be adjusted, as is the current can be adjusted as well. And in a power supply with a DC circuit, the voltage, current, and resistance are related by Ohm's law. So here you can see one way of writing out Ohm's law. It's voltage equal to current times resistance. And it's important to have an intuitive understanding of the relationship between voltage, current, and resistance, as that is helpful when you are performing a gel electrophoresis and setting your voltage and current. Importantly, you can rewrite this equation in terms of the current or in terms of the resistance. Obviously, you can write this as current equals to the voltage over the resistance, or alternatively, you can write it as the resistance equal voltage over the current. Importantly, to the right of the picture of the power supply that I just showed you is a picture of an electrophoresis chamber, and you can see that the wires that stem out of the power supply can be connected to the electrophoresis chamber based on the colors. So the black wire would go in here and the red wire will go in here. Importantly, you should understand the principle of a cathode and an anode. For example, the black wire that you see in the power supply will connect to the black spot here. This is where your cathode is, and your cathode is negatively charged. It will be the location where the electrons will leave. So this insert where you add the black electrode or the cathode will have a wire that goes across the electrophoresis chamber, and it will result in the release of electrons out of the cathode. On the other hand, the anode is labeled in red, and the anode is the spot where electrons will enter. So electrons will eventually move, and these electrons will enter the anode labeled in red, as you can see. And so this produces a full circuit where you have the electrons continuously moving this way, and that results in the production of a full circuit that can make gel electrophoresis possible. Importantly, in the figure that you see at the bottom is a schematic diagram of an electrophoresis apparatus. You can see that you have the power supply here. It connects with the cathode. This is where the electrons will leave the cathode and enter into your gel electrophoresis buffer. And you can see also an anode here, and the anode is where the electrons will eventually enter into, and they will enter the anode and leave the buffer solution, eventually coming back to the power supply, and then you will have obviously a full circuit that is allowing for the production of an electric field. So the whole idea of having the anode, the cathode, and the power supply is to generate an electrical field across your agarose gel. So in the figure, you can see the agarose gel here. This in blue is the agarose gel. And what you will have is an electrical field that is generated across this gel. Then samples are added into the gel wells. The gel wells would be somewhere here. In the picture on the top, you can see that the gel wells are somewhere here. And it is where you add the samples, which will be subjected to the generated electrical field. Importantly, before I move on, I want to discuss some important key points here that can help clarify matters, just in case there may be some confusion that arises if you look into the current and the electrical field from a physics point of view. So in physics, current is by definition the rate at which a charge moves. Basically, if you have a wire, say this right here is a wire, and you have a charge somewhere, you will calculate the current basically by understanding the rate or the speed by which this charged ion will move across a region. Now, by convention in physics, the directionality of current flow is diagrammed as if you are assuming that it is positive charges that move from a higher potential to a lower potential. So if you look at the particle that I drew in the wire, you can assume that this would be a positive charge if we are drawing the directionality of the current going to the left. However, this convention was created before the discovery of electrons, and it is actually electrons, assuming this is a metal wire, that would be moving and creating a current flow. I say this to you because this also is important when you consider the convention of drawing out electric fields. I'll get into that in a bit. But for now, I want you to understand that it is actually electrons that move in an electrophoresis chamber to create a full circuit. And this convention also applies to the directionality of an electrical field within physics. So when we discuss an electrical field, let me draw a simple electrical field for you. Let's say we have a positively charged molecule here, or ion, and we have a negatively charged molecule or ion here, and they produce an electrical field. In physics, the directionality of the electrical field is drawn out in a diagram as if you're dealing with the movement of a small positively charged particle. So for example, in physics, if you were to draw an electrical field here, the lines would be pointing this way, just like this, towards the negatively charged part. So this right here would be the electrical field, and obviously electrical fields are a little bit more complicated. They'll move like this and like this. But the idea is that the movement or the directionality of the arrow is depicted as if you have a positive charged particle that is moving in the electrical field. I say this to you because it might be confusing looking at figures where you are taking into account the direction of an electrical field across a gel electrophoresis. So for example, if you take a look at some figures of gel electrophoresis with an electrical field labeled, you'll often see that the electrical field is labeled as going from the top part where you add the gel wells into the anode. So this movement actually is depicting the movement of electrons and not the movement of a positively charged particle. And it's important to distinguish this from an actual accurate diagram of an electrical field within physics. So now that we have a basic understanding of the power supply and the electrophoresis chamber, we can get into the electrophoresis theory. So electrophoresis theory, as it is currently understood, depends on the following equation, where you have the V, that is the molecule's velocity, that is, it is the molecule that you add in the well. Obviously, you don't add a single molecule, you add a sample with many molecules. The molecule here could be a DNA strand, or it can be a protein sequence. So V here stands for the molecule's velocity. So obviously, when we're looking at the electrophoresis theory, we're looking at it as if we're singling out a single molecule within our sample. And so the molecule's velocity is dependent on the electrical field, or I should say the magnitude of the electrical field, times the molecule's net charge, that is the molecule or the molecule within our sample that we've added into the well, divided by the frictional coefficient. And the frictional coefficient is dependent on the molecule's mass as well as shape. So overall, you can deduce that the molecule's velocity is dependent on the electrical field that is generated between the cathode and the anode, as well as the molecule's net charge, as well as the mass and the shape of the molecule. By simple reasoning, we can deduce that the molecule's velocity is proportional to the electrical field and the charge. On the other hand, it is inversely proportional to the frictional coefficient, which depends on the molecule's mass and shape. Now, as I illustrated in the previous slide, what you will have is a gel. On the right side of the gel, say this is the wells of the gel, we'll have the cathode, and on the left side, we will have the anode. You will have an electrical field that will be generated across the gel. And it is the magnitude of this electrical field that is represented by E. As for the molecule's net charge, as well as the frictional coefficient, then this is dependent on the actual sample that we add. So this would be dependent on the sample that we add. And obviously, the velocity is just the product of the interactions between E as well as the net charge and the frictional coefficient. The velocity is obviously displacement over time, which is speed with direction. Now, in physics, when we are dealing with an electrical field and we're dealing with the movement of a molecule within that electrical field, then we classically consider the movement or represent the movement of a charged particle within the electrical field by mobility. This is the electric mobility equation that I have here at the bottom, and it's represented by epsilon equal to the velocity of the molecule divided by the magnitude of the electrical field. So if we want to modify this electrical mobility equation to adapt to our electrophoresis theory equation, then we can rewrite this as being the mobility or the electric mobility equal to the electrical field times the molecule's net charge divided by the electrical field times the frictional coefficient. So just to save you some time in figuring out how you get to this equation, I'll just rewrite the mobility here in terms of our equation on the top here. So the mobility would be equal to the velocity over the magnitude of the electrical field. So the velocity is equal to EQ over F, and this is divided by the electrical field. And so you will end up with EQ over EF. Importantly, this is the relationship of the electrical mobility as it pertains to our molecule's net charge, frictional coefficient, and the magnitude of our electrical field that we are applying. Now, keep in mind that oftentimes we are running a gel electrophoresis under constant voltage conditions. What that means is that the electrical field here and the electrical field here will be constant, and the electrical fields here will be canceled out, such that you end up with a ratio of Q to F. In other words, the mobility of our charged molecule that is moving in our gel is proportional to or is represented by the molecule's charge divided by the frictional coefficient. So in other words, this is a charge to mass ratio if we neglect to consider the shape of the molecule. So if the shapes of our samples are identical, meaning you are dealing with linear sequences of DNA or linear protein sequences that are not folded due to denaturing, then you would be dealing with a charge to mass ratio. In other words, under constant voltage conditions, the electrical field magnitude will be constant and the migration of molecules is dependent only on the ratio of Q to F, which is the charge to mass ratio in the case that the shapes of your molecules do not differ. So just to drive everything home, let's assume that this gel that you see here is within a gel electrophoresis chamber. It's covered in buffer solution, and on the top part right here, it's connected with a cathode. So let's say that here you have a cathode, and on the bottom you have an anode, and the cathode and the anode are connected with a power supply. Obviously, electrons will move out of the cathode, and eventually they will move through the buffered solution into the anode, and this will produce a complete circuit, assuming that here, for example, is your power supply. You'll have a full circuit whereby the electrons can move and create an electrical field. So the electrical field is running across the gel, and this electrical field will drive the movement of electrons as well as the movement of your molecules or samples which contain the molecules of interest. So for example, if we take a look at this sample here, it can be DNA or it can be protein. In the case of DNA or RNA, they're both negatively charged, so naturally they will move downwards towards the anode part. As for proteins, proteins would be treated such that they are negatively charged or coated with negative charges so that they also can move downwards towards the anode. As for the electrophoresis theory, our understanding is that the velocity of any of these molecules within these samples, obviously this band right here would not represent a singular molecule, it would be many molecules. For example, if this is DNA, it would be many DNA sequences that are in here of the same size. But as for the electrophoresis theory, our understanding is that the velocity of a molecule is proportional to the electrical field magnitude and the net charge of the molecule divided by the frictional coefficient. And if we want to write this in terms of electrical mobility, then remember this would be epsilon equal to the electrical magnitude times the net charge of the molecule over the electrical magnitude times the frictional coefficient. And you can simply write this as Q over F.